actually that is what I wanted to start off with, is a poem from somebody who lived in Maine and went to my high school. And Edna St. Vincent Millay, she also lived not far from here in New York. But she wrote in 1939 a poem that really captures very well the state of neuroscience today. In fact, the state of big data today. And she wrote, "Upon this gifted age, in its dark hour, rains from the sky a meteoric shower of facts. They lie unquestioned, uncombined. Wisdom enough to leach us of our ale is daily spun, but there exists no loom to weave it into fabric." I think that seeing this in 1939, she was certainly very prescient. And in fact, she captures very well the challenge that we have in neuroscience, the fact that if we bring the lights down, please, we're studying many different brains, right? We're studying mouse brains and rat brains and cat brains and monkey brains, all in the goal, ultimately, of understanding the human brain. And it's really, of course, the human brain, as was mentioned in the previous talk, and it's the human brain that we're uniquely interested in. It's what underlies who we are. And it's also what underlies many disorders and diseases that affect who we are. And this presents a particular challenge because we don't understand actually the pathophysiology of any of these diseases. We don't understand how any of the treatments that we found for any of these disorders or diseases actually work. 70 to 90% of these don't work in 50% of the population. And a tremendous number of the findings, actually biomedical findings, these are the findings that give pharmaceutical companies the lead, the direction to follow to develop new compounds and new drugs. 75% of these findings are not reproducible. So we've got a big challenge here. The pharmaceutical industry, to a large extent, is giving up on major new investments in the brain. And so that's the downside. But on the other side, we're getting a tremendous amount of data about the brain. So this is phenomenal. And there's more to come, as we see. There's the brain activity mapping proposal, the activity in the United States to come up with activities to generate much more data about the brain, Human Connectome Project, and so on. There's a lot of data to be produced. The technology is increasing almost weekly to generate more and more data about the brain. But the big question is how do we actually bring that data together in an integrated form in order to accelerate our understanding of the brain and ultimately the human brain? And that's really what the Human Brain Project is about. It's a 10-year European project to launch a global collaborative effort to understand the human brain, specifically enabling advances in neuroscience, in medicine, and in future computing. So it's one of 2 projects that were selected earlier this year for €1 billion in funding over the next 10 years. So it's a flagship, I'll explain what that is, and it was officially launched October 1st, so just a few months ago. It consists of a consortium of 256 researchers, 135 research groups, 81 institutions across 22 countries. So it's funded in principle for €1 billion over 10 years. Half of that is coming from the European Commission. The rest of it is in matching funds. And the first slice of that is €74 million for the first 30 months. And then there's regular reviews. So we haven't received suitcases of cash. A FET flagship is actually something, this is a program in the European Commission to drive technology innovation. So this money is not coming from traditional neuroscience funding. This is coming from an effort really to trigger in Europe large-scale initiatives that are risky, that are innovative, that are visionary, to derive new benefits for society and to keep Europe competitive. So that's, I think, important to know. And the goal of the Human Brain Project is really about laying the technical foundation for a new information technology-based way of doing brain research, to drive data integration and knowledge integration across different disciplines, and to trigger really a community effort to achieve a new understanding of the brain, new treatments for brain diseases, and new brain-like computing technology. So there are 3 key research areas in neuroscience where the goal is really to integrate everything we know about the brain into computer models and simulations. in medicine to derive new ways of understanding and diagnosing and ultimately treating diseases of the brain, and also to learn in computing to learn principles from the brain in order to develop new technologies that are building off of the principles. For example, the brain is probably the lowest power supercomputer we can imagine, and we need to learn how to build off of those principles for our next generation of supercomputers. So the scientific organization, probably this is far too small for anybody to see, but I'll just focus on the key points. There's a little bit of data gathering. Mouse data, human data, some cognitive data. But the focus is really on building informatics-based platforms. And then with some applications to test those platforms and demonstrate that they're scientifically usable. And the data to be generated is actually strategically selected data just to get us started. We're not actually a brain mapping project. And that's really critical. We're really going to generate some data, and the strategy for the data generation is to identify key data from the genetic level, from the transcriptomic level, proteomic, vascular, synaptic, and so on, in both mouse and human, that we can link together to start building what we call a data ladder to the human brain, which is basically identifying where is it that the mouse genetically, transcriptomically, is similar to the human, and where does it diverge? And keep organizing along that basis the data. But the key thing is the Human Brain Project is not primarily about generating data. It's really a data integration project, counter to what you may have heard. And the core of that, what that data integration process really is about iteratively building what we call unifying brain models. So this is essentially taking experimental data, taking the data from existing literature, taking existing models, integrating them through model building processes into models that can be simulated, run on supercomputers, analyzed, compared, and validated against biological data that was not used to build them, and to keep refining that so that through this iterative process, you're constantly finding where does each piece of data fit into a detailed biophysical picture of the brain. Now, that doesn't mean that you have to simulate all of that detail at the same time. You can pull out layers of that to identify principles and simplify it. But we want an integrated form, an integrated representation of the current data and knowledge about the brain. And that's really where theory is key. Because we know that you're not going to go and take a collection of measures and just throw them into a bucket and suddenly have a working brain model. Right? You need to have high-level theories of cognition and behavior, of learning and memory, of tissue organization, and use that to organize the way that you integrate the data, and use both high-level cognitive models and low-level data-driven models, and bring them together in the middle. That's the gap that we're missing right now. We know a lot of detail about the low level. We know we have a lot of theories about the high level, but we need to bring them together in an integrated model. So to do that, we're developing 6 platforms. And those are neuroinformatics, brain simulation, medical informatics, high-performance computing, neuromorphic computing, and neuro robotics. And the aim is for these to be open to the whole research community. I'll go very quickly through these platforms. I can answer more later. But the neuroinformatics platform is really about federating data from different data repositories from around the world, making it accessible to analyze structurally and functional data. So, for example, extract features from EM or analyze relationships within functional data, correlations in functional data. But the core principle is to organize that data using both space and ontology, semantics, into multi-level brain analysis, so that all the data is organized and queryable, searchable in a way that the brain simulation platform can actually make use of it to build models. And so this idea then is really collecting data, organizing it, curating it into registered searchable data, models, and literature. Now, the very controlled data, the data that's been gathered with specifically prepared protocols, that can be used for building models. But most data today is actually gathered using sort of ad hoc protocols for specific hypothesis testing. Now that data is still very useful for validating the models and for going in and testing ideas on the models. The brain simulation platform is really about providing a technical platform, an internet portal for neuroscientists to come in, go through a workflow of building up a model protein, a model cell, a model synapse, circuit, ultimately whole brain, and instrumenting it with virtual instruments. So, for example, simulated optical imaging, local field potential, put an electrode into it, virtual EM, and be able to link those simulations to a simulated body and a simulated environment so that we can have closed-loop in silico experiments. And the point of this platform is really to make it possible for neuroscientists to ask new questions and prioritize what would be the most useful next experiment to perform. So it's a new tool for neuroscience. It doesn't replace it. And the brain simulation platform will support many different representational levels, from individual particle tracking up to abstract morphological representation, up to even mean field, it's not represented there, volumetric representations, and full brain regions. And of course the mathematics to simulate that as well, from coarse-grained molecular dynamics, reaction diffusion, multi-compartment Hodgkin-Huxley, up to point neurons and mean field. And we have some experience kind of testing how to integrate data from the Blue Brain project. And this is a project that started in 2005 with a partnership with IBM. And it was started on the basis of a collection of data that was gathered over 15 years by Henry Markram in his lab. And so this was a collection of data including electrophysiological characterizations of individual cell types, reconstructed morphologies, paired reconstructions of morphologies, so identifying the properties of the connections between those cells, and also putative locations of synapses between these cells, and also extracting the cytoplasm and looking at the gene expression products in specific cell types, as well as characterizing the synaptic, the short-term plasticity and the long-term plasticity characteristics between them, and the composition, all on one single brain circuit. So it's all focused on somatosensory, primary somatosensory cortex at a specific developmental age in rat. So it's all same animals, same protocols over 15 years to a large extent. And using that data, so I came in 2006, and we had the challenge of, well, if you have gone through all of the painstaking work, you heard in the lesson, several weeks to reconstruct a single morphology. If you've gone through the work of gathering all of that data, what do you do with it? And we wanted to see, is it possible to actually use a data-driven approach to build it into a computer model that we can then simulate and evaluate how well does that data that we spent all this time gathering actually explain emergent phenomena that we can observe in the network. And so we started to try to capture the electrical diversity of all of these different cell types. And there's a broad number of interneuron classes that show many different firing types, and all of that diversity is actually created by a great diversity of ion channels. These are proteins that sit in the membrane and sculpt the response, the voltage response of an individual cell. And so we started off building models of lots of different types of ion channels, and we also built a project to start characterizing that using cultured cells where we cloned the ion channels. I don't have time to go into the details of that. But then the question was, okay, well, how do you actually build a model neuron? Traditionally, what had been done is if somebody was going to model a specific cell type, they got a morphology, they got some electrophysiological traces, they got all the literature together, and they sat down, and then typically it was a PhD student that spent 3 years to build a model neuron. But if we had to capture 30,000 neurons for one small piece of brain, that's not scalable, right? So we had to figure out, how do we build these automatically? And how do we make sure that it's all data-driven? It's integrating directly the data measurements. So we took reconstructed morphologies. We had information from gene expression to give us some hint as to the composition, not complete, but a hint of which ion channels should be in each cell type. We have the ion channel models, And then the question is, well, how are those ion channels distributed across that morphology in order to recreate the electrical behavior that we've been measuring? And so we use multi-objective optimization processes to distribute those ion channels across using whatever constraints we do know. So there is information from biology about certain characteristics of the distribution of these channels, and we use that information. And in the end, we generate automatically individual model neurons that recreate the observed firing types and generalize to other types of stimuli than what we built them off of, including network-like input, and also can be validated against detailed, these are dendritic recordings of calcium spikes, for example, in the dendrite, and we also could basically put in the objectives into this multi-objective optimization process from the data and get out models that recreate these phenomena. And so in the end, we learned how to build automatically hundreds of thousands of model neurons capturing all of the diverse electrical properties of both the pyramidal and the interneuron classes of neurons. This is the layer 5 pyramidal cell model, which was the focus of that publication. And then we went and we had representatives, of essentially examples of all of this diversity of morphological and electrical and genetic diversity within this circuit, and we start to build the model circuit using the data that tells us how many of each cell type are there with which density within this one volume of a cortical microcircuit. And this is just kind of a view on the inside to show you we're just placing these cells. And there's this tangle then of all these axons and dendrites. And the big question is, okay, so you're putting all of these together, but how do you know you're going to connect them correctly? Because of course you know the axon needs to be really close to the dendrite. And how are you going to make sure? These are neurons reconstructed from individual animals, all separate animals. These neurons didn't grow up together. So it can't possibly be right? We can't get the correct position of synapses, can we? And this leads me to an example of something we call predictive neuroscience, because we don't think it's going to be possible to measure every single aspect of the entire brain. So we're going to have to use the data that we do have and find out how much can we predict from it. And the fact is that within this simple cortical microcircuit, there are 2,970 possible synaptic pathways between all of the different electrical and morphological types of cells. And 22 of those have been fairly robustly characterized in terms of the detailed physiology and the anatomy. And the question is, can we identify some principles that help us predict the ones that haven't been measured? And so one piece of that problem was understanding, well, where are the synapses positioned? And we had data from 15 years of experiments with these paired reconstructions, giving us distributions of where are the synapses positioned along the dendrite and on the axons. And we thought that once we put all of these neuron morphologies together into the volume, we were going to have to kind of tweak and shape the morphologies in order to recreate those distributions. But we found out we didn't. Just simply the shape of the morphologies alone actually is sufficient to predict most of the positions of the synapses within that circuit. And so then that gave us insight, first of all, that most of them we didn't have to go and measure all of the detail, but there were exceptions to that we could then focus in on. For example, the chandelier cell is a famous interneuron where you need to actually have a more specified targeting mechanism. And so we can layer in those rules. And then, similarly, for the number of synapses per connection, this is an important parameter in determining the impact of a synapse. And we learned that by layering in additional constraints like Bouton densities and certain thresholds for the connections being sustainable enough, that we could actually predict not only the mean number of synapses per connection, but the distribution of those synapses per connection for the population. And similarly, now we're seeing that we can predict connection probabilities for many pathways. On top of that, of course, synaptic plasticity, shapes, and skull tolerance, but these are the structural foundations that are all the plasticity is built on top of. I'm going to have to get a bottle of water, excuse me. So then, if you put all of that together, this is an old simulation, but you put all of that together and you turn it on, Well, first of all, when we first turn it on, it's silent. It doesn't really do anything. Like a sliced little bit of spontaneous activity when we have the minis on. But then when we excited the whole thing by changing, for example, the extracellular potassium, then the whole thing starts to exhibit these low-frequency oscillations. And so what was interesting is that this was already something comparable, coarsely, to what happens in vitro. When you actually excite the whole bath, you change the bath solution in a slice, you can elicit these low-frequency oscillations in cortical slice preparations. So it gave us the clue that, although it's not perfect and it's not all exactly right, it was already in the right parametric ballpark only by putting together the low-level measurements, the low-level details. We didn't tune or select any parameters to reproduce a specific emergent phenomenon, but we looked at what does the data give us. And then we could use this as a tool then, the fact that we have this computational model, to actually go in and compute, well, what's the extracellular potential for each individual neuron? And if I turn it up, like here's some spikes. And so we're simulating, actually, what would you get if you stuck in a wire electrode? But we're simulating it for the whole volume. And what that means is we can now calculate from the model a local field potential and use that then to relate real local field potentials to activity of individual populations of neurons. So it gives us a new tool for understanding the signals that are being measured from the real biology. This is work that was done actually in collaboration with Christoph Paul and Process Anastasio at the Allen Institute. And so then we took that cortical microcircuit model and we replicated it many times in order to make a virtual slice. And we cut the fibers and made a 230 micron slice. And this is just showing the spontaneous activity in that slice, as you saw before in the calcium imaging from the real biology, but we have in the simulation, I don't know quite where this is. You were a bit stubborn. Okay. So here that you can see just the spontaneous activity in this simulated slice. And then we can take this and start to do experiments on it and continue to validate it and refine it. And for example, one experiment that we set out to reproduce and see if it would reproduce is actually from Rafael Yuste's lab, from Michael Bayerlein, where they saw that by stimulating at low frequencies and at a range of different frequencies, the cortex actually acts as a frequency selective gate. And layer 4 specifically, then with the right frequency of stimulation, which in their case was 40 hertz, they triggered polysynaptic activity. So we simulated thalamocortical input with a density of synapses into layer 4. It could be better simulated, but this was a start. And then we stimulated a bunch of different frequencies. I'll just show you a few here. We simulated at 20 hertz, and you get a little bit, so this was for a few thalamocortical fibers. And you get a little depolarization, a little bit of fire, and not much happens. But then when we stimulated at 40 hertz, watch what happens. So then we basically trigger this propagating recurrent synaptic activity. It follows the basic path of propagation that was also seen in their paper, spreading up from 4 to 2, 3, and then down to 5, 6. And so we're continuing to constantly refine and validate that against thousands of experiments, actually. And so it's a tool then for continuing to integrate our current data and knowledge about cortical microcircuitry. We also have work in progress, and this is in collaboration with a group, Pierre Magistretti at KAUST in Saudi Arabia, but we're extending the model at a small level to look at the coupling between individual neurons and glia and the vasculature, because this is going to be very important to understand that relationship for the energy consumption of neural activity and linking it to fMRI, the bold signal, for example. And we're also scaling the infrastructure from that individual cortical column or cortical microcircuit, scaling it up to be able to build the full rodent brain. So the other platforms, the high-performance computing platform, is really dedicated to building the computing power needed to run these simulations. And that the key capability that's new there is making computing and supercomputing interactive. We know as the scale of these supercomputers grows and grows, we'll no longer be able to write all of the data out to hard drives. Just firing up the hard drives necessary to write out all of the data that will be generated by a simulation will cost more and take up more energy than the computer itself. So the whole challenge is then, how do you change the way supercomputers are used to actually do online interactive simulation and interactive analysis so that the simulation is actually performed during the course of the simulation and the visualization. And this is just to give you an idea of the scale of the computational problem. From a single cell taking about a gigaflop, a billion floating point operations per second, to simulate, and then where we started in 2005 with a UGML format to simulate this neocortical microcircuit. And then as we moved up, we upgraded to a BlueGP to do these mesocircuits. And now we're at the level of, we just had a new supercomputer that will be capable of a cellular level mouse brain. So it's just one step below this rat brain. But we're going to need 1,000 times that to simulate a human brain at the cellular level. So that's not doing all the reaction diffusion detail, and all the molecular stuff, but just the level that you've been seeing for the human brain. And that turns out to be an exascale supercomputer. And what's interesting, and what was one of the motivations originally to start this project, is those supercomputers are on the roadmap to be delivered already. Not because of neuroscience, but because of large-scale simulations of nuclear bombs. But wouldn't it be cool if we could use that to actually understand something more about biology? And so we're talking as part of this project. We're in regular conversations with them, and they have partners that are developing supercomputers, but we're informing them about the requirements we have to do these simulations, so that, and they think that's kind of cool too. If it can do more than simulating a bomb, great. So they're actually taking into account some of our specific application needs. Medical informatics is a sub-project that's really aiming at federating clinical data at a large scale, very large scale, making sure you don't individually identify any patient, but do aggregate analyses of populations for lab tests, brain imaging, the diagnoses, and identify, basically by doing these queries, large-scale queries across hospitals, to come up with a data-driven definition of disease. And that will give then some specific ways of targeting areas for treatment, and ideally get away from the symptomology-based definitions of disease that we've been heretofore strapped with. So this is an effort underway. It'll start with clinics in Switzerland, and then expand from there. The Neuromorphic computing platform is really about building hardware to run simulations at very large scale, very high speed, and very low power. It's building off of two projects, two European projects, Brainscale, which has been doing large-scale, wafer-scale analog VLSI, and Spinnaker, which is a project that is actually from the inventor of the ARM chip. So every one of you that has a cell phone has an ARM chip or two in their pocket. And this project is using millions of ARM cores to run real-time simulations, digital simulations. And this is a key part of the high-performance simulation outside of supercomputers. And then, of course, the Neuro Robotics platform is really not about hardware robotics at all, but it's about providing simulated agents, simulated bodies, simulated environments that can be coupled in closed loop with the simulations of brain circuitry. So we know that behavior is essential, and we know that coupling to a rich environment and a body and sensory input is really essential. There are a couple, I don't really have time to go through all of these, but the potential applications, of course, understanding principles of cognition. If you've got a full simulation of an organism in a brain, you put it in an environment, it makes a decision, you can trace back through all of the things that contributed to that decision or that recall of memory. This is also a tool that can be used in identifying new drugs for treating brain disorders, and looking at what the secondary effects would be, and then also for developing new types of chips for industry, for cars, for example, or consumer devices. And then it's really critical, of course, that other areas be addressed. And there's substantial investment in ethics and engaging the public and bringing in philosophers, bringing in, forming an ongoing dialogue about the developments of the project and the export of technology and the issues involved in whole brain simulation. Education is really key. Dissemination, governance, I have no time to discuss those things. I just say, this is the organizational chart. But we don't want that to be static. So 20% of the funding has been set aside for open calls in every funding cycle to bring in new partners, only for new partners. And I encourage you to go to the website to learn more or ask me questions.